I'm Dr.Riyaz. I'm the founder and CEO at Centella AI. I come from a drug discovery background. I'm a medicinal chemist by training. Medicinal chemist is the one who is the fulcrum of drug discovery. Whenever the drug discovery is there, a design, chemistry, biology component comes. So I stand in between, understand the design component and biology component and the chemistry component. So that's my with my decade and a half experience in drug discovery. And then in 2021 we realized that the drug discovery industry, especially the preclinical drug discovery industry, is moving from an era of knowledge driven to informatics driven to data driven. Hence, we realized that integrating the knowledge and as well as the informatics driven drug discovery into data driven will accelerate the drug discovery. Hence, we started Central AI Therapeutics in 2021 to accelerate preclinical drug discovery in specific through human first and machine next intelligence. And then we have been, funded by multiple people, the ISB and Government of India and angels as well. And as you know that the drug discovery is very, very effort intensive. It consumes 10 to 15 years of time and huge cost. And then you know that 90% of the clinical trials failure. So the idea here is to make the preclinical drug discovery much faster, so that you have the pipeline of the funnel of clinical trials is more. Even if you fail at an 90% level, still the faster the therapies can come into picture. And this is the typical value chain of drug discovery, which starts with a typical target discovery, wherein the protein which is responsible for the disease you have to identify and then validate whether it is the right target or not. And then you have to find and hit the molecule which goes and binds. It's a lock and key theory. You will be identifying a lock, and then your job as a drug discovery scientist is to identify the right key that opens the lock. And then you have to identify lead and lead optimization, then do preclinical trials on animal. And then once you prove the safety efficacy, then you move towards the clinical trial phase 1 to phase 3 and then FDA approval. uh, Again, coming back to preclinical drug discovery, whenever let's say breast cancer, the BRCA protein, if you want to identify a new drug or discover a new drug, then you need to design a molecule that goes and binds to the protein and make them in chemistry laboratory and test them in biology, analyze the data. So this is what you call it as in medicinal chemistry context, or DMTA cycles: design, make, test, analyze. And this is an iterative process, wherein in traditional setting you have 20 30 iterations going on, and you end up synthesizing 3000 to 4000 molecules to take one molecule to the clinical trials level. However, this is where the major time cost gets consumed. Now we want to use our expertise from traditional drug discovery as well as informatics and AI, and reduce the iterations to less than 10 and reduce the number of molecules you synthesize and test to less than 300. So with that intent, we built our platform called Cascade, Centella's AI supported computational discovery engine, to accelerate hit and lead. And as a medicinal chemist or as a drug hunter, whenever a target is being given, a protein is being given, I have two challenges as to what molecule should I design and how do I synthesize. So there are around 10 AI workflows which are integrated into Cascade, and these are some of the workflows which help us, design or throw ideas. The generative AI role here is to throw ideas or build ideas which are beyond human imagination. As a medicinal chemist, I don't need any tool. I just need the protein information, the previous clinical trial, drug structure, and the IP space, and then I can draw the structure or design a molecule. And also have a virtual screening which can screen up to 1 billion molecule, and the absorption, distribution, metabolism, toxicity prediction tool which can predict up to 50 endpoints. And once you design the molecules, that's a great job, but eventually you have to synthesize them in chemistry laboratory. It should be feasible. And we have a synthetic, retrosynthetic tool which can predict multiple steps and this vendor availability, and in how many steps you have to check, and what is the cost of that raw material per gram. Whenever you do design, you should be also aware of the synthesis and also the scale up. Mg level, it's wonderful, but when you go for a ton level, when you go for phase 1, phase 2, your synthetic molecule should be scalable and you should have a root and raw material available. So this is a typical architecture of our entire platform, which is now converting into agentic AI, entirely prompt driven. And we have two patents on our platform on the pocket coordination molecule generation on the entire platform. And we work with Microsoft Discovery. Microsoft Discovery is working on a specific project for agentic AI. And also this platform is entirely human and lab in the approach. All said and done, you designed a nice rubber in the laboratory, but it has to meet the road. The rubber has to meet the road to understand the efficiency of the tire. So we used our platform to do an proof of concept of one of the targets, which is CDK2, which is overexpressed in hormone receptor positive cancers, triple negative and ovarian cancers. Now we wanted to use this target and design molecules using our platform and then test them in real time in wet lab. So this is a typical protein signaling pathway which plays a vital role in cell signaling, or like cell in a cell cycle process, I would call. And what we do before we allow the AI to jump in picture, we do our due diligence from the medicinal chemistry point of view. That's where our human drug discovery expertise comes into picture. The due diligence of the target, like what is the target, whether it is druggable, is there any clinical trials available, is there any FDA approved drugs are available, and what are their structures, and then what is the chemotypes that are available, which is it benzene, pyridine, or like benzimidazole or indole, what are those key structures available, and then what are their activities there. And also we also take into the account of, is there any failed clinical trials, and what is the structure of those failed clinical trial drug, and what are the structural attributes that are contributing to that failure of the clinical trial. So this is the typical due diligence what we do, and then followed by, we also understand the IP space. The drug discovery is all to do with the IP. The larger IP blanket you have, the better it would be, so that no other player enters into the domain. So we do a lot of IP analyses, like which companies have patents on the specific target, what are their structures, what are their structure activity relationship, and based on that we should actually curate this specific structures data. And also we do CDK2 is an it has got multiple isoforms, and we need to be very selective about CDK2, because the reason why we have chosen selective CDK2 is for the factor that CDK4 and 6 already FDA approved inhibitors are there in the market, which are hyperactivating CDK2 and leading to the resistance. Hence we wanted to block the CDK2 and going forward have a combination of CDK2 plus CDK4 and 6. And we realized that a specific amino acid residues are responsible for the selectivity of CDK2. So that's where we identify what are those, and based on this data we curate the information from the open source and then fed to our algorithm of structure based drug design, wherein we give the target pre-DB structure, and the algorithm reads the pocket coordinates and the atom types and the geometry, and based on that, this binding site where the molecule, this is a protein and this is a binding site where the molecule go and bind, and based on that the algorithm reads what are the pockets, atoms and types and geometry, all those nitty gritty which are needed to generate drug like molecules. And once you do that, it generates molecule, and once the molecules get generated, sometimes it generates the junk also, or majority of the times the AI generates the junk. That's where the medicinal chemist role comes into picture. What filters to apply, what to filter, what not to filter. So that's, let's say if it is an onco drug, there are separate filters. If it is a neuro or CNS drug, there are separate filters you have to apply, so that the right set of molecule comes into picture. As you know that signal to noise ratio, always the noise is more, the signal is less. So similarly in generative AI, the noise is more and the signal is less. Now you have to smartly identify, apply filters to remove the, weed out the noise. And once you do that, you design molecules and you have to check whether they are synthetically feasible or not, based on the how much will be the overall yield, the number of steps, and then eventually we actually go for an synthetic and experimental validation. So these are some of the molecules or chemotypes which the AI has generated. We further validate them whether they are in sync with the previous literature, or is there any IP overlap, and followed by which molecules are binding in the hinge region of the protein, and what are their binding poses, how they are interacting, are they interacting with the reference drug molecule with the same amino acid residues or not, and then followed by, this is how we shortlist from 900 molecules to the design to apply filtering, and then eventually 40 molecules in chemistry laboratory synthesized, and then check the biological activity. And this is how the hit rate, we had in 3x improvement hit rate from a traditional context, and then time and cost being approximately reduced to 50%. And we have an very extremely encouraging data. We were looking for an hit like molecule, we landed up in lead like molecule with an 7.3 nanomolar activity in enzymatic studies, and this is the FDA approved drug, so it is almost close, you can say. And also carried out some MTTSA studies wherein it is some of the molecules are extremely 52,000x selective towards the cancer cells versus normal cells. Whenever we design a molecule, it should only go and hit the cancer cell, but not the, and leaving intact the normal cells. So if you see the CTX-C2A3 is very selective and then showing 21 picomolar activity on cell lines and with 52,000 selectivity. Now this is a 10x improvement the existing Pfizer molecule which is there in clinical trials. So ideally we plan to have a phase 1 in next 18 months. And this is another case study in rheumatoid arthritis, and then we also have similar results, and 7x improvement and 50% reduction in time and cost, and much better than the reference drug, and 188 nanomolar. And we work with Structural Genomic Consortium Canada, and we also there's an open call. Around 70 companies applied globally, and 5 have been shortlisted, and we happen to be one among them. And this is a first-in-class target. And also we work with University of Catanzaro on one of the metabolic and NAFLD target, and then we are applying a joint patent with them for an European patent. And these are some of the success story how we helped some of our clients across global, some of the academia, small mid biotech, and then helping them to reduce the time and cost by 20-50%. There are diverse domains of mostly small molecules and in ADCs, antibody drug conjugates. So this is our core team, myself, who's a medicinal chemist by training, and Dr. Puna, he's a cancer biologist. He comes with close to 12 years of experience, and then Mr. Jeevan and Drew are our BD partners and strategy. And we have an advisory board from Harvard Medical School and University of Catanzaro, Albert Einstein College of Medicine, and example. We are a true interdisciplinary team, wherein medchem, biologists, informaticians, AI engineers, and data scientists, and we do have. So we see drug discovery through the lens of AI, but not the AI through the lens of drug discovery. So on this note, I'll be happy to take any questions. Thank you very much for the opportunity.
Good morning. Excellent talk, Dr. Riyaz.
Thank you.
I think so. The session is open for questions. Before that, I have one question for you.
Yeah.
So it's excellent technology, and I have also moved from the basic researchers. We have done a lot of innovation. Some molecules we have discovered, and being from India, we always believe that how we can do something re-engineering and try to find out a way how we can avoid the patent, and we can become the first generics. So some of the molecules I like to highlight, like pegaspaginase, which was just becoming off patent, and there was only one company who was selling that injection at around 2 lakhs rupees in our country. We did reverse engineering, and of course all the clinical trials and all, and we launched that product somewhere around 25,000 rupees. So there's the power of what we have. So one question to you, because we believe in AI so much. So when we shortlist a candidate, and may I request everyone to put on the mute? So we have to do the activity on the cell lines. So is there a methodology you have worked something? So still we have to do your studies on the cell line, or is there a way how we can mitigate that and find out some shortcuts so that even the cell line studies we can reduce?
Yeah, thank you for your comments, doctor. Two points I want to make. That AI is doing wonderful, that's fine, but you're telling like you have to trust or believe AI, but my statement is don't trust AI. Don't believe AI, okay? Believe human first and then machine next. The ingenuity comes from your, your experience, basically, how you guide it, how you, do it, number one. Number two, hence is the reason why we select Central AI, accelerating drug discovery through human and machine intelligence, not machine and human. Point number one. Point number two, in terms of cellular activity, and then, see, AI is not a panacea. One drug treating all diseases, and it will not happen, number one. Number two, replicating the complex biological system environment through AI is extremely complex, doctor. Though there are companies, Meta, tried, burned human, good amount of money, Chan Zuckerberg initiative, there are umpteen number of companies. But I'm not skeptical or cynical, but next 1 decade is going to be a good amount of improvement. But the challenge still lies in how do you replicate the complex cellular biological system through AI? Because it's very dynamic, extremely dynamic, as the medical oncologists know, like whenever you give any chemotherapy drug, like the pH, the environment, and then how it passed through, there are umpteen number of factors which influence. So I don't say I'm very cynical or skeptical, but I see that's an area which needs more, resources, more in-depth basic research that can predict, replicate a near true biological system or cellular system.
Any comments from you, sir? Or
Maybe genomic replication or interpretation.
They are there. See, that's in genomic replication level, and then anyways, the real time experimental data has to get digitized, and then it has to be evaluated. But now the genomics data is being widely used for the target identification. I was talking to Dr. Arijit. Majority of the times, AI predicted targets are not faring well. Not faring well. Even if you see the AI drug discovery companies across globe are working on the targets which are well explored, at least 80-90% of the times.
Even the regular animal studies, usually they may not be more than 20% successful in human trials.
Yeah, yeah.
But the replication of that biological environment may play a good role.
Yes.
Actually speaking. Okay.
Yep.
Thank you.
Thank you.
any questions from the audience?
Yes.
Hello, Dr. Riyaz. Thank you for your talk. It was wonderful. I have worked in preclinical and clinical both, so I can understand that the time taken for this humongous process in like finding out the lead molecule. So my question is around that. Using an AI, how much acceleration of this drug discovery process coming to the lead optimization you have seen here, and how much of this 40 molecules which you arrived from the 900 molecule screening was converted into a clinical trial?
Thanks for the question. That's a very pertinent question. We have not yet moved to the clinical trial level. We are at a preclinical lead optimization. Ideally, it starts with hit discovery, lead identification, lead optimization. So that each phase you optimize certain parameters. It's like a DJ music system where you will be, optimizing multiple knobs to get the right, experience of music. So the moment you up one parameter, the other parameter goes down. So you have to strike a balance. Number one. Number two, in terms of we are currently at a lead optimization phase where the in vivo studies for the CDK2 target which we have shown, and for the other two targets, and then the target on autoimmune, and then for the metabolic disorder, we are already validated in vivo in mice. So I think in the next 12 to 18 months is somewhere we are looking for our phase 1.
Hi, Dr. Riyaz. Good
Hi.
to see you again.
Thank you.
What challenges are you facing right now with respect to bringing it to clinic? Because you have been operating in this field for quite some time, right?
Right.
What challenges are you anticipating and facing in this field? Because so far everything is good with respect to labs and your core, right? The challenge will come when you would really like to convert it to a product, right? Where we are not good at as an ecosystem. Any thoughts on that?
Yeah, that's a very good question. Ideally, we keep talking about deep tech, right? So when we started, we are a deep tech company. But later we realized that we are not only operating in deep tech space, but we are operating in deep science space. So ideally, we are operating in a space of deep tech and deep science. A typical deep tech to come into the ROI model takes generally 7 to 8 years. Okay, but it has shrunk to 5 years now. And typical deep science also takes 5 to 7 years. But what we could achieve is the POCs and then getting into commercialization or like making the buck, showing our product that has already been validated. The major challenge is now TRL 4 to TRL 9. Now we are at a TRL 5 approximately in terms of 7 in terms of technology, but in terms of science we are at a TRL 4. Now the valley of death starts. You need huge resource, huge investment to convert this, in vitro data into in vivo data into clinical data. So that's where a major challenge is going to be. A huge investment, dedicated investment in specific the patient capital is needed.
Yeah. I'll also add, see, there are definitely several challenges, but we'll have to work on a platform from the traditional ones to the latest one, which is AI aided. Now, AI is not going to replace everything. If we are saying at one strike of button, everything you get the data and everything will be goody goody, it is not going to happen. But yes, what AI is going to assist a lot of things. Like traditionally, earlier we were screening so many molecules. So that funnel has reduced. So now we can work on the selected target. That is first. Second, my question was like, if we, from there, we want to generate some in vitro data. Now generating in vitro data itself was a massive task. Because we have so many lead candidates, so you have to work on what is the absorption, what is dissolution, what is the metabolism, so many things are there. So that can be substantially reduced by AI because they can predict. But it can filter. So from say 100 molecules you filtered at 50, and from then 50 you filtered to 25. And then when you go to cell, again still you'll have to do that. So AI can predict you helping something. Now there are some molecules which AI has predicted and they are into clinical. So it is going to funnel down, correct, but it's not going to replace. I remember old days when somewhere in late 90s, when the whole government wanted to establish, want to make computerize everything, if you remember. So there were strikes, that there were so many employees on strike, railways and everything, that all the jobs will go away, everything will go away, so many strikes. What has happened? Nothing has happened. So computers in place, we have advanced. So similarly here, this is a transition. Every era has a transition. Now this is an era of AI, so nobody has to fear that everything is going to go away. It's going you to guide, and for that guide, ultimately your human's minds are there. See what happens to bacterial agents. The virus and the bacteria are so smart, you discover a new molecule, they will change themselves and the mutation will happen. And you will have to, and that's what you have to apply human minds. So AI is going to assist you, it's not going to replace you.
Very much, sir. AI is an enabler, accelerator, but not a replacer, in summary.
Anyone more last questions? Yeah, last question here.
My question is regarding, have you employed any explainable or interpretable models to understand and verify why a candidate has been passed and why a candidate has been rejected through the screening process?
To a certain extent, for me, explainable AI is through human validation. Like there is a still amount of a sort of black box. Like why it is generating the way it is generating. Though you can give some sort of logic, rational justification, but still there is certain amount of gray area. Now that can be filled with human intelligence.
Yeah, thank you so much. Give a round of applause.
Thank you, doctor.
Thank you so much.
Thank you so much.